Okay, let's go over in this video apoptosis initiation and how it works. So first, why do cells apoptose? Number one, embryogenesis. It's needed to, create space between fingers, toes, etc. And once tissues aren't needed, to go away. Number two, it's used in the immune system all the time to stop an immune response. Once the pathogen's been cleared, we don't need the PMNs anymore, so they can apoptose. If there's not a second signal, it probably demonstrates that the cell is recognizing self, it's lost tolerance, so that cell should apoptose, etc. And then also if the cell is infected with something and another cell thinks it's infected, or if there's just damage to the cell so that it might be causing mutations, etc., the cells will apoptose. And the reason that they apoptose and undergo this kind of coordinated and energy-dependent process is because cells want to avoid necrosing. Necrosis is when cells just kind of die too rapidly. They blow up, all their cell contents go to the interstitium and then into the blood, available for pathogens to scavenge, to also cause damage to other cells. So we apoptose to prevent that. Two major pathways to apoptosis: the death receptor extrinsic pathway and then the cell stressor intrinsic pathway. The extrinsic requires an exogenous signal for the cells to apoptose, hence the name. Intrinsic doesn't. The cell senses that it's stressed. Either there's no survival factors coming from the interstitium and the blood, or there's DNA damage, etc., and the cells undergo a cascade of events to have them kill themselves and die gracefully, basically. Just briefly, intrinsic pathway, it has more steps than the extrinsic pathway, and this kind of makes sense. It follows other cascades where the intrinsic cascade is longer than the extrinsic because when you get to the extrinsic, there's already been kind of regulation upstream. So once the exogenous signal's there, it's probably a true signal. Versus with the intrinsics, cascades, etc., you want to prevent just kind of aberrant, one protein bumps into another and then activates the cascade. So you want more coordination, and thus it's longer. And then the other final thing is that both of these require energy. That's key. So just to briefly introduce the players of what's going on, caspases are the major players. Caspase stands for cysteine aspartic proteases. They're the major effectors of the pathways. Once you get to caspase activation, you're pretty much committed to apoptosis. And then there's the regulators, and the regulators are different based off the pathway. Extrinsic, again, is fast ligand, fast fad, only 3, which is less than the intrinsic. These are just listed here. We'll go through them in the next few slides. Okay, so the extrinsic pathway, as I said, is easier to think about, and that's because the production of fast ligand is itself very regulated. So once you get there, things are ready to go. And what happens is fast ligand is a ligand expressed on CD8 positive or NK positive cells, natural killer cells, and it binds to FAS, which is a transmembrane protein of the target cell, induces FAS to trimerize next to itself, which then enables the recruitment of FAS-associated death domain, FAD. Once that happens, FAD goes ahead and cleaves procaspase 8 into active caspase 8, which then goes on and activates the major executioner caspases, mostly caspase 3. Caspase 3 is the major executioner caspase of apoptosis. And in fact, in labs, etc., in order to figure out apoptosis activity, they have readouts of caspase 3 activity because they're synonymous. Once you have caspase 3, you will apoptose. So that's the extrinsic pathway. Pretty easy. The intrinsic pathway is much more complicated. First, just think about what's normally going on in a cell. There's normal survivor signals. These enable the production of proteins, in particular BCL2, which stays on the mitochondrial membrane and prevents cytochrome C leakage. Right, so just like this shows right here. No leakage to cytochrome C. Now, just as a side note, do you recall which cancer has BCL2 overexpressed? And by overexpressing it, cytochrome C can't leak out, which is a critical thing of apoptosis, so it blocks apoptosis. The cancer is follicular lymphoma. It's the most common non-Hodgkin cell lymphoma, just to go over. And it's the result of BCL2 being put downstream through a translocation of chromosome 14 to 18 downstream of the IgH promoter. And that's actually how BCL2 gets its name. It stands for B-cell lymphoma 2. All right, so that was how normal cells normally prevent apoptosis. BCL2, again, is expressed on the mitochondrial membrane, prevents cytochrome C from leaking out of a pore. Now, with apoptosis, what happens is the cell, which used to have some survival signal, now either lacks that, or there's radiation or whatnot that causes DNA damage. The cascade of DNA damage is pretty interesting. So first, what DNA damage does is it turns on a very important gene and slash protein called ATM kinase. ATM kinase, being a kinase, what it does is it phosphorylates different targets, and for apoptosis, the targets that are most important are MDM2 and MDM2 and P53. By phosphorylating MDM2, what it does is it inhibits MDM2, and phosphorylating P53 and activates it. MDM2 normally, its normal activity is to constantly grab P53 and target it for proteasomal destruction. It's the E3 ligase of P53. So when you block and inhibit MDM2, then you have more P53 levels because it's not getting destroyed. And by directly phosphorylating P53, what that actually does as well is prevent P53 from being bound by MDM2, and it enables it to translocalize to the nucleus where it can act as a transcription factor. In particular, P53 turns on a whole array of proteins, but in this system, the ones that are important are BIM, BID, and BAD. These are also called BH3 only proteins because they associate with the BH3 domain of BCL2, and they only can carry that domain, hence BH3 only. I underline BAD because I think that's maybe the most important one of these three. But what they do is, as the little cartoon shows, is that these proteins right here, they go ahead and bind to BCL2, and when binding to BCL2, they prevent it from preventing cytochrome C from leaking out. And actually, too, they enable this Bax channel, which is also getting expressed by P53, to not be blocked by BCL2, and then enable cytochrome C to leak out even more. So that's how they kick things off. And just as a side note, you recall which virus has its own MDM2 analog, and thereby, by having an analog, it's breaking down P53 and blocking apoptosis. Well, the virus, if you don't remember, it's HPV, and in particular, HPV's E6 protein. That's how it works. It's basically a viral MDM2. So then you always have P53 blockage, and you don't get induction of apoptosis. Okay, so now let's start just from the top. Once you have cytochrome C leakage, what that does out of the mitochondria, what that does is now it's in the cytoplasm. It enables the conformational change of APAF1. Okay, APAF1 is this really neat protein that's normally kind of kinked on itself, and it's a closed formation. Cytochrome C comes along, and with DATP going to DADP, so again, it's energy, requires energy, it causes the unspooling or the opening up of APAF1. APAF1 then goes ahead and sees itself, and with some more energy, results in this beautiful wheel shape called the apoptosome. And it really is beautiful, might I say. And it's the center of APAF1 is the hub of it, and then the spokes are APAF1 with cytochrome C bound. Okay, so the point of this apoptosome is to then enable further recruitment of caspases. So just kind of moving up to the top right here, the apoptosome then recruits procaspase 9, which sits on the top of the apoptosome, and that in turn, with that sitting like that, enables the recruitment of caspase 3 to come along, and it cleaves procaspase 3 into caspase 3, which is activated now. And again, caspase 3 is the major executioner of apoptosis. So once you get caspase 3, you're committed to cell death. And it's thought, well, why do you need this whole apoptosome and unspooling and coordinated thing? Well, the thing is, these are all pro-proteases that then have to get cleaved to become active. And in order for that to happen, you need them to be, and they all cleave each other, right? Procaspase 9 cleaves procaspase 3, etc. So in order for that to happen, you need them to be co-localizing in space and time, and that's what the apoptosome does. Also, by not having the apoptosome around normally, the chance that these randomly will bump into each other in the cytoplasm enable a kickoff of this cascade is very, very low. So that's how the cell protects itself from, apoptosing when it doesn't need to. All right, so that's the two pathways in general. Should be noted, though, that they do intersect slightly. Of course, it's always more complicated than it's initially made out to be. So procaspase 8, which again is the key of the fast ligand, fast FAD, extrinsic death receptor pathway. Once it gets activated, normally, as this figure shows, it then goes on and activates caspase 3 mostly. There's also 6 and 7, which then cause apoptosis. However, caspase 8 can also go on and work on one of those BH3 only proteins, BIT, to activate it, and then cause the intrinsic pathway to go. And also, just I like this figure because it shows that if you remember T cells, CD8 positive T cells and NK cells, a lot of them, beyond fast ligand way of killing cells, they also express perforin and granzyme, which is basically they insert a channel in the membrane and then insert granzyme. Well, I think one, the perforin alone with that channel causes huge leakage of things out of the cell and calcium leakage into the cell, which causes huge damage and cell death. But two, as this figure shows, granzyme also activates this BH3 BID protein, which causes the cell to undergo intrinsic pathway-mediated apoptosis. So kind of interesting how they do intersect. So in summary, the big points are extrinsic is shorter, it's fast ligand to then fast associated death domain. It works through caspase 8, that's big. Then the intrinsic pathway is longer. It first causes DNA damage or lack of survival proteins, etc. So the cell becomes stressed. This causes activation of P53, which results in blocking BCL2 activity in the mitochondrial membrane. Without BCL2, cytochrome C is able to leak out. And it's also, there's an expression of a protein which facilitates this as well. When cytochrome C leaks out, it enables APAF1 to unspool and to form the apoptosome with itself, which then enables caspase 9 to then activate caspase 3, causing apoptosis. And then the final point is that while these are usually distinct, they do intersect. So that's that. Hope you enjoyed it. Hopefully it's useful.